Hello everyone, welcome back to Biology 102. We are going to begin with the cell and its parts. And this is just a short video to say hi, but I don't want to use up file space, so the rest of the slides will just be voice recordings. Before we begin, let's review the rules of the class. These should be familiar. Will Nisar Aleo, I'd like you to please read these and have students explain what each means. And you can pause the video. And here are some tips on strategies to study. Pause the video while a student reads and explains. This is how the various parts of this class will be graded, just like in other classes that I teach. And here is a chart that you can use to keep track of your grades. Please notice that if you do poorly in a couple areas, you can still make it up by doing well on the intra exam and final exam. This is how you can calculate your grade, so there should be no surprises at the end when you receive your final grade. Let's begin with the lesson. For our next class meeting time, I want you to be able to draw and label the parts of a plant and animal cell. So practice this in your notes. These are the topics we will cover. Some may be review, and all of it builds on what we learned in past lectures. We'll look at cell size and ask, why are they so small? How do we see cells? What do various types of cells have in common? What is in the prokaryote cell and what is in the eukaryote cell? We'll look at animal versus plant cells, major organelles, and the plasma membrane. We saw this slide in a previous lecture. It's important to many areas of science to understand the concept of surface area to volume ratio. How do little things get in and out of a cell? They go through the membrane. The more membrane a cell has, the more stuff that can go in and out. But how does a tiny cell create more membrane? By having greater surface area to volume ratio. So look at these two examples of a sphere with a large volume, but it will have relatively small surface area. And we have a sphere with a small volume, but a relatively large surface area. Another way to think of this is through the example of cubes that you saw in a previous lecture. As the area increases, in this example, the total volume remains constant. Thus, the surface area is increasing compared to the volume, and more things can go in and out through the surface. Let's use a real-life example of an elephant and a mouse. Which one has the larger volume? But which one has more surface area? Well, the elephant has both larger volume and more surface area than the mouse. But let's look at the relative surface area to volume. Small volume, but large surface area for that tiny volume. Which one will lose heat more rapidly? The little mouse will lose heat more rapidly because it has more surface area for its volume. What do we use to look at cells? Microscopes. These are only two of the many types of microscopes. The simplest is this bright field microscope, which merely allows light to pass through the sample and we can see it in its natural color. This is probably what you have in your lab. There's another type called an electron microscope. It allows examination of the fine details. In this example, we have tracheal cells. So let's get to the part we really want to study, the cells and differences between prokaryotes and eukaryotes. Give me some examples of prokaryote cells. You can pause and discuss, and also give me some examples of eukaryotic cells. So for prokaryotes, you might have said something like bacteria, the archaea, or blue-green algae. For eukaryotes, we have plants and animals and fungi. Knowing the differences between the types of cells will help us when we need to treat diseases caused by prokaryotes versus eukaryotes, or to treat the cells themselves. A bacterial infection, which is a prokaryote, is treated much differently than a fungal infection, which is caused by a eukaryote. Let's begin with the 5 things that prokaryotes and eukaryote cells have in common. They all have plasma membranes that defines the cell and holds in the internal structures. Cytosol is a semi-liquid substance. When we consider the organelles, or organites in French, with the cytosol, we call this cytoplasma. We also have ribosomes that are found in all cells. These organelles make proteins. We're going to learn about this more in detail. And finally, chromatin, which is made up of DNA, or ADN, and proteins. And this is visible when a cell divides. So these are 5 things that prokaryotes and eukaryotes have in common. Here is a prokaryote cell. So we can look for these 5 parts. I would like a student to come up and explain the parts. So pause the video and then play it again. And did you get this right? The plasma membrane goes around the cell. All cells, including this prokaryote, have ribosomes, which are tiny structures that make protein. There's DNA that is combined with protein to form the chromatin. Cytosol is just the liquid in the cell. But when we consider all these little organelles with the cytosol, we call it cytoplasm. And notice where the DNA is. It is not in its own membrane. This is a very important difference. So please watch this video and look for the parts that we have discussed. Now let's look at eukaryotic cells. Here we have an animal cell, and this is the vegetable cell, the plant cell. We'll look at the things that it has in common with prokaryotes. We see the cell membrane in both. Here's the cell membrane of the plant cell. Cytosol is the liquid that's floating around inside. But if we consider the little organelles and the liquid, we call it what? Cytoplasm. So the plant cell also has this liquid and the cytoplasm. The DNA, this is very important, the DNA is inside a nucleus. Here's the nucleus. And a difference between plant and animal cells is plant cells have cell walls. And this makes them more rigid since they don't have skeletons. Let's look into more details about the organelles that we find in eukaryotic cells. We'll talk about these 5 groups, starting with the nucleus. And I want you to remember what eukaryotes are. You should be able to name the kingdom that each of these are in. And a hint, this is over here. This is supposed to be Jean-Jean. So, what kind of organism is Jean-Jean? And these other ones are from previous lessons. You should know what they are. We'll begin with the nucleus, which contains the instructions of the genetic code. Inside the nucleus, we also find the nucleolus, which makes ribosomes. The nucleus is surrounded by its own membrane called a nuclear envelope. This is a phospholipid bilayer similar to the plasma membrane that surrounds the entire cell. Remember phospholipids from the previous lecture? Here is a diagram of the nucleus. The nucleus is the purple structure, just to help you see it. It's not really purple. It has a nuclear envelope or membrane that surrounds it. This membrane allows things to travel in and out of the nucleus through these pores called nuclear pores. And you see all these little red dots? They represent the organelle called the ribosome. So there's many ribosomes on the membrane. They're also on this blue structure that we will cover in more detail. This blue thing is the endoplasmic reticulum. Inside the nucleus is this thing called the nucleolus. This is where ribosomal RNA is made. We'll talk about RNA and DNA replication in future lectures. The RNA takes the DNA and turns it into proteins by using the ribosomes. These are the ribosomes, these little red dots on the membranes. They make proteins for the cells. Without ribosomes, we would not exist. We have free-floating rhizomes that float in the cytosol of the cell. We also have attached ribosomes that's on these membranes, including the endoplasmic reticulum. Here is an electron microscope photo of a pancreas cells. It shows both free-floating ribosomes. It also shows ribosomes attached to the endoplasmic reticulum. Remember that the ribosomes make protein. So if a plant or animal can't synthesize a certain protein, it might mean something is wrong with the ribosomes. This is a photograph of the pancreas cell, and it's the pancreas that produces insulin. Here is a diagram of the ribosome itself. It's actually made up of two parts, a large unit and a small unit. What is the endoplasmic reticulum? It's a membrane attached to the nuclear envelope. Remember, that's the envelope that surrounds the nucleus. The endoplasmic reticulum, in English I would call it the ER. In French, do you say RA? It manufactures and transports molecules. There are two types of ER, a smooth ER and a rough ER. The smooth does not have ribosomes. That's why it's called smooth. It manufactures lipids and metabolizes drugs and alcohol by breaking down those molecules. It also stores calcium ions for muscle function. The attached ribosomes produce proteins that are transported to the exterior of the cell. These proteins tend to be ones that are secreted from the cell, like insulin. Here's another look at the ER with the rough ER. What does it have? What are these? They're the ribosomes. And the smooth does not have ribosomes. The ER also has closed cavities that we call cisternae. So see these cavities? These cavities can break off and form these sacs that we call transport vesicles. They carry their contents to something called the Golgi apparatus that we'll talk about next. The Golgi apparatus adds sugar molecules to the proteins that are inside these sacs, and then the Golgi carries the contents to the cell membrane and can eject it from the cell or secrete it. The Golgi apparatus is also known as the cell manufacturing and shipping center. So let's pause this video and have one volunteer read this description, and then another volunteer explain the 6 steps described in this illustration. And then you can go to the next slide. And here is more self-directed learning for you. This is very important information to have once you get into treating animal diseases and learning how animals function. So you'll need, or you should need to know this for future classes. So pause the video while you have one student explain the illustrations and another can read the text but I want you to discuss it so that you understand what is happening. We're moving on to two more organelles that are important in the transfer of cell energy: the chloroplast and the mitochondria. Animal cells have only mitochondria, but plant cells have both chloroplasts and mitochondria. Also, some protists have chloroplasts, and most, but not all, protists have mitochondria. So let's examine the differences. Chloroplasts are the site of photosynthesis. They convert solar energy into the chemical energy found in sugar, as you will learn in the lecture about photosynthesis. Mitochondria, however, function in cellular respiration. They convert the chemical energy in sugar into energy that our cells can use. So here is a basic illustration of the cycle of photosynthesis over here by the chloroplast and cellular respiration by the mitochondria. Chloroplasts use the sun's energy to produce carbohydrates that the mitochondria then use to give energy to our cells. In the process, carbon dioxide and water are produced by the mitochondrial respiration. And then this carbon dioxide and water are used by the chloroplasts to continue photosynthesis. So plants have both chloroplasts and mitochondria because they both make food and they use that food to power their own cells. People, animals, only have mitochondria. We have to eat plants in order to get the carbohydrate energy. The cytoskeleton is the skeleton of the cell. It is made up of protein fibers that spread throughout the cell. It functions to hold the parts of the cell in place. It allows movement of intercellular substances. It keeps the shape of the cell, and it facilitates the movement of cells, such as muscle cells. There are three types of cytoskeleton: microtubules, microfilaments, and intermediary filaments. You will learn about all three. Here's the three types again: microtubules, and we see both a microscopic picture of it and an illustration to help us visualize it. Here's the microfilament, and finally intermediary filaments. So I would like you to pause again, have three students volunteer to explain each, and we see the protein, the structure of the protein, and the functions of these types. So please stop and explain these. And finally, we have the plasma membrane, which surrounds the cell, keeping all of the internal contents together while preventing unwanted things from entering the cell. How does it do this? Well, we have seen this picture in the lecture about the macromolecules. We learned about lipids and the double phospholipid layer that comprises the membrane. So we have two layers here. On one end is the hydrophilic head, and in the center here are the hydrophobic tails. So please stop and show all of these videos, and hopefully you recognize the structures of the cell. We're going to move on to looking at proteins in more detail. We learned about them in the previous unit, but now that we've learned more about cell structure, we can review the functions of proteins with all this new knowledge in mind. So we can have 6 categories of the functions of proteins, and we're going to do more self-directed learning by having students explain the next slides. Please pause the video and have 3 volunteers explain each of these 3 types of proteins. 3 more volunteers explain these proteins, and now you should become more familiar with the parts you are seeing, like the plasma membrane and the cytoskeleton. And finally, this illustration summarizes each of those 6 types of proteins. Please look at it and point out the 6 types and what they are doing. Here are questions. You should know the answers for future quizzes and exams. And finally, I have some homework for you. I would like everyone to get yourself into 8 groups, and each group picks a different organelle. So only one group picks the nucleus. One group looks at chloroplasts. I would like each group to create a 1-page sheet, only 1 page. So we're going to keep this brief. Write your names on the sheet, draw the organelle, give me a 1-paragraph summary of the functions and products of the organelle. And finally, a couple sentences about why this is important for agronomy. Why do you have to learn this information? And next week, you are going to present this information in class. You will have only 3 to 4 minutes. You will be graded on 4 things: the time, how long your presentation is. If it's less than 3 minutes or more than 4 minutes, points will be taken off. So you need to practice. We want to see everyone has participated in some way. Presentation has to be organized and easy to follow. And Wilnice and Elio will watch your presentations. Maybe record 1 or 2 for me, or parts of them. And they'll be grading based on these items. So, I often say half of life is just following the rules. So if you can just do these 4 things for your presentation, you will get a good grade. If you do just these 3 things for your paper, you will get a good grade. Remember, only 1 page And you must include a drawing, explain the functions and products, if any, of the organelle, and why is it important for agronomy. So thank you for coming to class.